There are three typical programs in PCR procedures: denaturation phase, annealing phase and extension phase. Firstly, let's begin what happens in denaturation phase. The double strands of DNA is denature at 94 Celsius for 2 to 5 minutes. While, in annealing phase, the temperature variable for a specific set of primers, so, this phase is the most critical one for getting a successful PCR reaction. On the other side, extension phase, generally be at 72 Celsius. This is the operating temperature for many thermostable DNA polymerases. So, the number of cycles depends on the number of copies of template DNA and the desired amount of PCR product. Generally are 20 to 30 cycles insufficient, as mentioned in the inserted figures in the next slides. In details, the double-stranded template DNA is denatured by heating typically to 95 Celsius. Then, the reaction is rapidly cooled to an annealing temperature to allow the oligonucleotide primers to hybridize to the strand's DNA template. Usually, annealing temperature is between 50 to 60 Celsius. After that, the reaction is heated to a temperature, typically is 72 Celsius, for efficient DNA synthesis by thermostable DNA polymerase. All theses details steps presented in the diagrams in the next slides. So, for a single cycle, denature DNA sample to separate DNA strands at 94 Celsius in 5 minutes, then primers bind to DNA strands according to its melting temperature for 30 seconds. After that, polymerase synthesizes new DNA strands at average 72 Celsius for 2 to 5 minutes, and so on in the next cycles, as mentioned in the diagrams inside the slide and next slides. This slide showed how the procedures of PCR amplification be done. In the first cycle, as noticed in the diagram, there are 3 steps. In the step 1, the double strand of DNA is heated to separate strands. In the second step, hybridization of primers be done, and between the second and 3 steps, the DNA polymerase, and DATP, DGTP, DCTP, and DTTP, be added to finish the DNA synthesis, from the used primers. In the step 3,